Okay, so sorry about that. We'll try and get through this in a hurry so people can have their tea. So I'll just be giving you a quick introduction to Oxford Nanopore sequencing and just sequencing in general. Like Lindsay said, we're reporting these sessions. So what is sequencing? So basically, let me just go through that. Yeah. So basically in sequencing, you're trying to determine the genetic makeup of the organism that you're looking at, right? So there are various methods that you can use: Sanger sequencing, next generation sequencing, or Oxford's nanopore technology. So by the end of this discussion, we would have gone through the basics of all of those. So basically, what is sequencing, right? Sequencing is determining the genetic makeup of the organism that you're looking at. We know all organisms, their DNA is formed of A, T, G, and C. If you're looking at organisms like viruses, which is RNA, it's AU, G, and C, because it uses uracil instead of thymine. So first genetic sequences were obtained in the early 1970s, and obtaining these sorts of sequences in the techniques we use these days involve both the wet lab, so performing the experiments, and also when you get the data from the experiments, knowing the bioinformatics tools and how to use them to then generate these sequences. So there are many uses of sequences. Like we were saying before, you can determine the relatedness of organisms if you know their genomic sequences. Sorry.
That's the word I went to get. But there should be another thing up anyway.
Oh, okay.
Yeah. Just
look at weather back to Okay. So you can determine the species of something. You can determine the relatedness of different organisms to each other. You can identify mutations that confer different characteristics to organisms. For example, in bacteria, we're talking about mutations that confer resistance. So using sequencing, you can determine all of these things. Can also be used in surveillance. For example, now, as we had the example from Dr. Solo, where he was talking about having to determine where someone has, contracted a specific bug from. So you can determine whether the bug was passed on from the mother, or whether, if you get all the samples from the community, you can kind of determine transmission patterns from how related the organisms are after you sequence them. So one of the landmarks of genetic sequencing was when we managed to sequence the whole human genome, right? This was like there was two groups that were planning to do the one-use shotgun sequencing, which is what we use these days. It was Venta's group, right? Venta's group did shotgun sequencing, which is what we use these days, which is we sequence fragments of your organism, fragments of the DNA of the organism, and then overlay the bits of sequence that are related to each other to build up the full sequence. So things mainly Sanger sequencing. So in Sanger sequencing, you separate the reactions into four. So you've got your ddNTPs and your dNTPs. So your ddNTPs, basically you've knocked out an oxygen off the ring. So once that ddNTP is incorporated into the sequence, you can't add anything after it. You can't add on more nucleotides. So you have the your enzyme add on ATGC, and then, of your deoxyribonucleotides. And then once it adds in a DDNTP, the reaction stops, right? So what happens that you get fragments of different lengths. And then those fragments, because the DDNTPs they fluoresce, that fluorescence is detected. So you have, your 2 base pair fragments, 3 base pair fragments, and all of these will then be taken by a capillary jolt of electrophoresis. And then the ones with the shorter fragments obviously will migrate further. The ones with the larger fragments will migrate shorter. So then you get like a sequence, basically, of DDNTPs that goes through from 1 base pair to however many base pairs you're looking at. It's 1,000 base pair fragments, right? And then those DDNTPs will then let you know exactly what you incorporated at each of the steps. So this is basically just showing exactly how right? So you run the reaction in 4 different tubes. In one you add DDA, dideoxy, nucleotide adenine, and then the guanine cytosine, and then you get something that looks like at the end you get something like this, right? Where all the dideoxy, and then you build up your sequence. So that there, C, T, C, G, then A, T, G, T, C, A, G, G, and that would be the sequence that you got, right?
So this is just showing what you just saw before, but in a video.
So it's just showing you have the enzyme, you add your sequence, now you're adding both DDNs and these to each of the chains.
In biochemistry,
So that was just the
to be
basic
able to determine
run through of Sanger sequencing. So while standard sequencing, you're only like sequencing a single DNA fragment at the time. NGS was sequencing millions of fragments, and then we build those fragments back up in by overlapping sequencing, and the overlapping will then be done afterwards using your bioinformatics, right? So there are two main platforms that are used for NGS sequencing. Illumina, which uses more shorter reads, like you were saying, from 35 to 600 base pairs, Keith? 300 on each side, right? Yeah. So you're not reading the full, you're reading the two halves. And Oxford Nanopore, which is that longer read sequences. So Illumina, like we said, sequencing by synthesis. These are short reads. The DNA is cut up into 200 to 600 base pair chunks, and you read it from opposite ends. So in the 600 base pair chunk, you read 300 from this side, 300 from that side. And then those are then the little fragments that you will then overlap to build up your full sequence. The DNA is amplified, so you get lots of those chunks of DNA, and then denatured and fluorescently complementary base pairs are attached. The fluorescence of the different colors is what's recorded, and that's what gives you, and that's how the sequence is identified. So your amplified DNA then attaches to your plates, and then the little fluorescents, which are shown over there, the fluorescent dideoxynucleotides are then attached, and that's how the sequence is read. So it will keep on attaching until it reaches either according to the length of the fragment that you wish to amplify, whether it was 150 base pairs long or 300 base pairs long. It will keep attaching those fluorescent nucleotides, which are then read by a laser at each step. And then that's how the sequence goes up. So then after you get those sequence readings, you will then use bioinformatics to then overlap complementary strands. And then that's what will build up your full sequence.
Sample preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as the sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The three ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Then the second flow cell oligo forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is washed away. This entire process generates billions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments.
That's why if you've ever done Illumina sequencing, you'll always get a read 1 and a read 2 because you get the paired end reading, so you get both the forward and the reverse strand. With Nanopore, it's different. You only get one single strand currently.
Yeah. Yeah, so like she was saying with Illumina, you're reading from either end of the strand, and then you're using those overlap sequences to then build up your genome. Whereas with Oxford Nanopore, we just either amplify the DNA that the strands that we're looking for, and then those strands are then pulled through a pore, as you will see later. And then the signal that's generated when each nucleotide goes through the pore is the one that's read off. So A will produce a particular electrical signal, a C, a T, and a G will produce their own specific electrical signals. So I think we can, did we go further? Yeah, okay. So yes, so Oxford's Nanopore Technologies developed the MinIM, or for sequencing of organisms in more remote, inhospitable environments. So you can carry like the MK1C, which don't even need an internet connection to read off. Sequences have been done, like we've shown in the Antarctic, in the Democratic Republic of Congo during COVID. During the COVID outbreak, lots of sequencing was done in really remote regions because you can just take that little like MK1C and the flow cells, just a little cartridge, you see it later on, and you use it later on, that you insert into it. You don't need, there's no need for a lot of power. Lots of these like carry battery packs, And so you can use it in regions that are, so where you don't have a huge lab where you can run your sequences from.
It's also a lot cheaper to buy a MinION than it is to buy, say, a high seq. So
And
the
yes.
starter pack for MinION is $1,000.
Yes.
What's the high seq going for these
Oh,
days?
gosh.
The cartridges, I think the cartridges are like 20,000, right? The cartridges is between 12 and 20,000 for like one cartridge.
For the sequence cartridge,
Yeah, the sequencer cartridge is the big one. Yeah.
for my C,
50,000, something like that.
yeah.
It's like
yeah,
50,000,
but-
right?
Yeah, but-
Yeah. So if you're doing a whole lot of sequences, like a whole lot of samples, then an Illumina kind of makes sense because you're spreading that cost over like a larger number of samples. But if you're using a small number of samples, for example, like we've got like 12, right? 12 barcoding kits and 96 barcoding kits, you would kind of use an Oxford Nanopore. And Oxford's Nanopore technologies, we use long reads. So whereas Illumina, it's like short 600 base pair reads, we do like thousands of base pairs. So the DNA library, you prepare your DNA library and proteins are added. These proteins, as you'll see later, attach to the motor protein and then enable the DNA strand to be pulled through the pore. And the nucleic acids are then like, so the proteins attached to the motor protein and the nucleic acids are pulled through the pore. And then the nucleic acids, as they're being pulled through the pore, elicit an electrical signal that's then decoded. So like I said, an A will produce a specific electrical signal, a T will produce its own, a C, and a G. So that's just showing you what happens. So basically you have you'll see when you get your flow cells, there'll be a little array. And on the array, that's where you have your reader and motor proteins attached to your array. So you put in your sample and your sample will wash over the array, right? And so basically your DNA with the proteins attached will wash over the array. The motor proteins will then attach to the fragments and pull the fragments through. We'll have the fragments will be barcoded. So when it's going through, the barcode will also be read. And then that helps you know which sample that particular read belongs to. And once you get the fragments from your various samples, those fragments are then overlapped when you use your bioinformatics to then build up your consensus sequence that you're going to get at the end. Yeah, definitely.
So the difference between nanopore technology and Sanger and Illumina is that Sanger and Illumina is sequencing by synthesis. They are actively adding DNA to your strand you want to find out about. Whereas Nanopore, you are just purely sequencing that strand specifically.
You would have already either amplified it or, you know, as we're going to do now, we did a culture and then we extracted the DNA from the culture. So the DNA that we got in the culture is what's been, so we're not like using an enzyme to add sequence to it. So there are different library preparation kits and sequencing platforms can be used for different things. So this one before, this one is the MinION, and we've got some, yeah.
It's like flagship one.
Yeah, so the MinION, basically, you just connect the cartridge to it, and then you connect that to then your desktop or your laptop, and that's what will be what will have the software that will run your MinION and like run the sequencing, right? Whereas with the MK1C, which Monica, you've got one of these, right? So with this, everything is integrated into that one platform. So you basically just start the sequence from you put in your flow cell into this slot here opens up. That's where you put in your flow cell. And then you use the interface there, which is on the screen, to start your sequencing on. And then so this is what it is. So your flow cell will go into here, into this platform. Then you just close it and then you interact with the interface here. And that's what will run the sequencing for you. Whereas with the little MinION, it's just this. So you get your flow cell, insert it there, connect that to your desktop, and the desktop is the one which will have the software called Nano that will help you run the sequencing experiments. Sorry. Yeah, so like I said, so basically if you are going out into the bush, you'll probably use an MK1C, right? You know that you're going to be doing most of your sequencing in the lab. You can use a little MinION, yeah. So the MK1C is a bit more expensive. I think it's $5,000
or $5,000.
$4,000 or $5,000. And then the little MinION, I think it's about $1,000 for the.
You get kits and flow cells with that as a starter pack.
And you get a start, yeah, exactly. You get a starter kits.
Considering one flow cell is I think £800 now, so it's that like maybe $850. It's pretty good value, really, I think.
Yeah.
The whole thing is $1,000.
Yeah, so if you're buying the flow cells on their own, after that, it will cost you $1,000 for one. I think when there was COVID, they reduced the price to $600, but I don't know if they still have that.
It's only if you're doing COVID.
It's only if you're doing COVID. Yeah, okay. So if you're doing other organisms, you may not get that discount, but yeah. There's also the Flongo, which it's the same technology, similar array, but you can do fewer samples on that one.
It's got a smaller array and less pores. And so if you're only doing, say, 2 or 3 samples, then it makes it cheaper. So instead of wasting a flow cell by only running 2 or 3 samples when you could be running 12 or 24 or whatever, it just reduces costs if you're doing small amounts. And it fits into the MinION as well. So it's not a different platform, it just kind of sits on the top.
And then you have the GridION, which is like a bunch of MinIONs run together. So with this one, you can do hundreds of samples. Basically.
It's also got a really powerful GPU computer in it. So it's an integrated
system
platform. Yeah, it's also quite expensive now. You're talking IC prices, I think.
Yes, exactly. And then Promethean is the same thing, right? It's just a bigger.
Has different flow cells.
Yeah. Oh, it's got different flow cells.
Tiny square flow cells, so they fit more on, I think.
So this is just like showing you the different platforms. So and like what the starter packs are. So yeah, GridION and Promethean, that would be like for a much bigger lab. But I would say probably MK1C and MinION is what most of us would use. and what I think the government bought most of during the COVID outbreak for sequencing, to enable sequencing in our countries. So, when we're doing sequencing, there's what we call depth, right? And then coverage. So depth pretty much means that at a particular position, for example, position 10 in your sequence fragments, right? You want to know if it's an A, T, G, or C that's there, right? So when you get a reading, it will either be A or let's say T, right? At that particular position. If you've got like 10 fragments that have aligned to that position, right? And that all say A, right? You have more confidence that that's an A. Whereas if you only have like 2 fragments that then say AA, right? And then you don't have any other information, you are less confident that at that position you got an A, right? So depth just means how many reads at that particular position do you have, right? So if it's 100, then it's a depth of 100. If it's 10, then it's 10x depth, right? Usually for TB sequencing, what's the minimum? I think it's 7x, right? At a certain position.
So with Illumina, because it's got the paired end reads, I think traditionally the sort of minimum cutoff is 30 times coverage. So you would want to get an average of 30 reads per say.
Or especially for the sequence, the bit of sequence that are repeating.
Yeah.
Right?
But with Nanopore, because traditionally it's slightly less accurate than Illumina, although that is now changing with the new versions of these kits. Traditionally, the minimum depth is 40. But if you're working on something like TB, which has it's very slow growing, it's very slow mutating, and as Keith said, the difference between the same organism and a different organism is down to sort of 10 or 12 base pairs in an entire genome. The better your depth, the more confident you'll be that actually what you're seeing, and obviously what you're seeing is such a tiny representation of the whole genome. You need to with TB, again, because it's a slightly more complicated organism, the better depth, the bigger the depth, the better. But I think the minimum cutoff for Nanopore is generally considered to be about 40 as an average across the whole genome. So it might not be 40 for everywhere.
At every position.
Yeah.
Okay. So, and then coverage is just like how much of the sequence you got, right? So once you finish running your experiments and you've got your different fragments and they've been aligned to the reference sequence, how much of that reference sequence have you covered, right? That's basically the coverage. So you get a reading once you do that, once you align your sequence of either 97% or 98%, and you have gaps in your sequence to show you the gaps where you didn't get enough information at those positions. So that's basically what it means by coverage.
Sometimes as well with MTB genome, where you see gaps, it's not necessarily that then repeats over them. Sometimes you have got these large deletion events, which can happen in your sample, where that whole region, even sometimes 2,000 base pairs will have been lost.
So this would be in the organism itself, like that.
So for example, when you look at your Beijing lineages,
yes,
at information 29,000, you'll find that there is a 10,000 base pair, 2,000 base pair region which has been lost in all the East Asian strains with respect to H37RP.
So those are like
so like your Beijing will have that one, and then your Central Asian one will also have another
deletions.
You definitely find them as both regions of difference.
Yeah.
So you also find that also happening in your
sequencing.
not critical typing. For critical typing, you looking more at the space and sequence of which have been lost. But this one is just like one contiguous piece of
DNA,
which
yes.
has been lost. So typically as it's when you have some mistakes happening and then
Yeah,
the whole issue happen. Sometimes there can be duplications whereby instead of the whole region being lost, you find it's
duplicated.
in
So I'll just give you like the basic and the way the flow cell looks, the anatomy of the flow cell. So that's the array. Yeah. And then this where it says spot on, that is a cover for the array. So when you pull it off, you have a little like for there that you then add your sample to. So then that's when the sample pressure. The array. There is also a priming port here. Is that? No, that's the priming port cover.
Yeah.
The priming port is over here, yeah. So the priming port cover covers the priming port. And once you move into the side, you have a port that you can then you add your priming solution that's supposed to wash over, because they when they store the array, they store it with the storage solution, right? So we'll have to wash off that storage solution while preparing the library and the sequencing part. So to wash over that, we use the priming ports. So that's where you add your priming solution. And then you have vent ports. These are the ones that will be used for waste. If you're going to pull out waste from the cartridge, because the waste will collect in these channels. Yeah. And that is, yeah, that's basically it.
Yeah,
We'll go through, yeah, we've got a video that will go through that with you when we're loading the flow cell. Because you need to prime the flow cell and then load it. Yeah.
one flow cell or something, one something.
No.
It's a multiplex. So you
Yes,
add barcodes
yes.
to each of your samples and then you pull them together. And when the sequencing, when it goes through the core, and if you have base calling on at the same time, so you can make the computer work out the ATGs and Gs and Cs at the same time. If it does that, and if you have that on, it can read the barcodes at the same time, and it will give you like a histogram of how many reads have gone through based on which barcode. So it can do it in, unlike Illumina, I think, it can tell you in real time how many sequences you get through your barcodes. With the 19.6 kit, it was designed for doing sort of targeted or, you know, amplified PCR based items. So you can put, in theory, 96 different barcodes onto it. But if you're doing whole genomes, somewhere between, so with CB, because as you'll find out, it's a bit more tricky, somewhere between sort of around 12-ish. But if you're doing something more simple like E. coli, then you can put sort of, we put 24 on and got pretty good depth for all of them. Potentially you can probably put more on. We just need to kind of experiment with that.
Yeah, and if you're using viruses, so like for COVID, you can put 12 on easily. And then you also notice that you can also reuse the flow cell. So if you're doing, for example, like TB, which has like a huge genome, usually you can only use you can only use the flow cell once, right? If you're going to get all the data that you need. But for example, if you're doing like a virus, which usually has very small genomes, you can reuse the flow cell. So we found for COVID, if you run 12, you would usually run it like for 4 to 6 hours, basically, and you have enough data, and then you can stop the run. Then you can wash your flow cell, add your storage media, store it, and you can use it like maybe 4 or 5 times. So depending on what organism you're sequencing, you can kind of gauge your costs, like how much it's going to cost.
You should in theory get X amount of reads out of any flow cell, depending on how many active pores are there. So you have to consider when you're starting, right before you even start doing this, you might want to consider how long the genome is or what it is you want. So if you are just looking for, say, 16S, or you're doing whole genomes, if you're looking for, like with TB, 4 megabases, that's obviously quite a lot, but also you want to know how many reads or what depth you are aiming to get. So you have to kind of calculate, and also in the protocol, we put the rough calculations, and you can kind of work out roughly how many you should be able to fit on a flow cell based on how much information you want for each of the things you're doing and how big those things are in the first place. So it's a little bit of kind of calculation, but it's the same with Illumina as well. You have to calculate that you have enough pores or you have enough space to actually get the depth you need. If you put too much on, then you're just not going to get enough information, and it's kind of pointless because you can't tell if it's accurate or not.
There are also different kits that ONT cell, and what you'll be using is the rapid barcoding kits in this library preparation. There's also the ligation kits. They also have various PCR based kits, and they have like the automated. So when you have like a machine doing your library prep, you can also get those automated kits.
So that whole tracks that you can see on the screen is an automated. It's able to prepare your library kit, your library preparation just automatically. So there's no hands-on kind of time.
Okay, so this is the basic workflow. So you extract your DNA or RNA from the sample. You prepare your samples for sequencing. So like I said, so for this, because we're using TB, we got it from culture. So we're just extracting the DNA from there and then going straight through, right? For other organisms, you may do a PCR, and then from the PCR product, then you do the sequencing. It will just depend on what organism you're sequencing from. So yeah, so in our case, we're doing it from culture. So we'll just we've already done the CTAB extraction for you guys. So you get the already extracted DNA. So you get your DNA, you prepare your sequence, your library prep from the sample that you got. Then you run the sequencing, and then obviously you get your signals, which will then be transformed into the sequence of your fragments. So then the fragments, you then use bioinformatics to clean up your fragments and assemble them into a whole genome. So your fragments will then be overlapped as to where they complement each other to create the whole genome. And then you then use downstream processing to create things like phylogenetic trees, like he was showing you, the tree that shows you how related these samples are and whether they cluster, so you can know, the relatedness of the samples that you sequenced.
All those sections in blue were the bioinformatics or the computer side of things. So it's just kind of showing you, actually, if you are going to become sort of a sequencing person, you're going to have to kind of hopefully learn some of the bioinformatics, because it's at least half and half. It's very much kind of spanning the two sides. You don't necessarily also have to assemble the sort of minimum bioinformatics, which hopefully we're going to try and explain to you on Friday, is base calling, because sometimes for whatever reason you might want to not base call at the same time when you're sequencing. If your computer isn't very powerful, then you would turn base calling off to allow it to sort of concentrate its efforts on sequencing. So learning how to base call so you can get the FASTQ files and then kind of concatenating it. So putting all the FASTQ files for one isolate into one folder, and then you can kind of do what you need to do with it downstream. So those are kind of the very basics.
Yep. So the minimum, so for as she said, all of this, right, actually from when you're base calling basically to like the end when you're constructing your phylogenetic tree, that's all the bioinformatics side of things. So you're going to need a computer with some basic specs. So Windows 10, Linux, Ubuntu, or if you're using a Mac, Catalina, I think is the lowest one now. And then you're going to need 16 gigabytes RAM or higher. I would say here, I would say starting from Intel i9. It's not like I
think basically the best computer you can possibly afford,
yes,
because it will, sequencing is increasing and improving so much so quickly that your computer, if it's a minimum spec one you buy now, it will probably be obsolete in a couple of years and it won't be able to keep up with your sequencing, especially with the new versions, the Q20 Plus kits. It kicks out. So I did one run, a metagenomic run, and it kicked out 200 gigabytes of data. So it's very much buy the best you can afford, because otherwise you're just going to have to buy another one in a couple of years' time.
exactly. And you might want to, when you're buying your computer, if you need to sacrifice, so you can buy, like, it's easier to buy external. So here we say the minimum storage for the external drive should be 1 terabyte, right? That can be your minimum, and then you can buy like additional storage drives. But you want to focus on your processing power, like having as much processing power in your GPU and your CPU as you can get.
That RAM.
Exactly. So as much RAM as you can possibly get. Yes.
How about 16 most?
Exactly. Yeah. So mine is 16 as well, and I can do like viral genomes easily enough. But once I get to TB, it's too slow. So this one that is here is a 128 gigabyte RAM. So that one can do most things. So, and the price you're looking at, I would probably think $2,000, $3,000, $2,500, $3,000. If you're looking for like a desktop, if it's a laptop, it might be a bit more. But yeah, that's around what you're.
The organization is well funded.
Yes. A server will be the best. Yes, so that then it can handle all of that. Yeah.
They're actually having. So what it was like $25,000.
Yeah.
$20,000. 84 gigabytes of RAM and 44s and. Yeah, like Xeon.
Exactly. So then, so everyone can use it when they want to run. Yeah. So.
But for soccer.
Yeah.
Chance, I think you would speak with them. I think it's massive. I think it's 780 gigs of RAM. Yeah. So I think also in terms of sequencing, the like.
They can give you like remote access to the server, like such can like upload jobs.
Now, so I'm putting in. I think they're starting with viral genomes in terms of the software to analyze, but we are helping them with
the only problem with a server is you have to have decent enough internet to be able to maintain that connection. With Nanopore, with Illumina, you don't have to have internet connections either. With Nanopore, certainly at the beginning, it clings to the Nanopore servers. So to begin with, you definitely have to have decent internet. We ran a workshop in Tanzania, and it took me 6 attempts to start a run, and there were people running around trying to find hotspots. It was chaos. So yeah, I learned the hard way that you definitely need very good internet at the beginning, but actually, I don't think it needs permanent internet. I think it just periodically pings. But you might find that if you have very bad internet, you might find the run might stop.
Yeah.
So for Nanopore, I think it's the computer. And also if you're saving the data, you should only save the data onto the internal C drive or internal drive, because again, if you lose the link to an external hard drive or a cloud-based thing, then you're going to lose all of your data. So we have always been recommended to save directly onto your C drive and then move it out as soon as you finish. So you've got another terabytes worth of data for your next run.
Yeah.
And also, as we were saying, you get so much data from these runs now that actually where you store it is a really important consideration. If you have, as I say, if you have hard drives, I've got about 4 or 5, 2 or 3 terabyte hard drives. I'm using them all up. So we at UCL have research storage drives, and I've got about 8, and they're all again 2 terabytes, and I'm using them all up really quickly to store this data. And it does start to add up as well, especially if you save all of the raw data. The FAST5 files are really big. If you just save the FASTQ files and get rid of the FAST5 files, that's obviously smaller, but base calling gets better over time. So if you go back to those FAST5 files and rebase call or base call high accuracy rather than fast base calling, you get much better or more accurate data. So it depends what you want to do with the data and whether you need it kind of further down the line. So storage and the cost of long-term storage is also really important because you can very easily rack up terabytes worth of data after a few runs.
So, any questions before we have some tea? No?
Everyone
right.
just wants tea.
Yeah, everyone wants tea. Let's go have some tea.